Hi everyone, myself Pragati. Now I am going to discuss about contamination, spoilage and preservation of egg. First, composition of egg. Egg are most nutritional and natural food. It considered as complete food due to their balanced composition of proteins, lipids, vitamins and minerals. It contains three main parts. There are shell, albumin and yolk. It contains specific composition and that functions. First, eggshell. Eggshell forms the outer protective covering of egg. It contains 9 to 12 percentage of total egg weight. It composed of calcium carbonate about 94 percentage and small amounts of magnesium carbonate, calcium phosphate and organic matter. It contains thousands of microscopic spores that allows gaseous exchange. It covered by a thin outer layer called cuticle. It prevents from microbial entry. Its functions are protect internal contents, prevent physical damage, act as barrier against microorganisms. Next, albumin. Albumin is also known as egg white. It consists about 58 to 60 percentage of total egg white. It is clear and fluid consists and surrounding the yolk. Water consists approximately 88 percentage and protein approximately 10 to 11 percentage. Minerals are small amounts and fat negligible. In their functions, supplies water and protein to develop embryonic, protects yolk by cushioning effect, act as natural antimicrobial barrier. Next, yolk. Yolk forms 30 to 33 percentage of total egg weight. It protects the egg portion. Water consists approximately 48 to 50 percentage. Lipids approximately 32 to 35 percentage. Proteins approximately 16 to 18 percentage. Fat soluble vitamins are vitamin A, D, E, and K. Lipids consist triglycerides, phospholipids, and cholesterol. Their functions provide energy and nutrients, supplies essential fatty acids, and support growth and development. In this picture shows about the outer layer part is shell and the center layer is thin albumin and calaxa and thick albumin. The center yellow color layer is called yolk and outer is vitelline or yolk membrane and the center hole is all germinal disco or blastodisco and then the outer layer of shell membrane and shell membrane present. Next topic is contamination. Contamination is presence of harmful microorganisms, chemicals or foreign materials contains in egg. It make unsafe for human consumption due to their porous shell, rich nutrient content and highly susceptible for contamination. If improper hygiene practice are not allowed to during protection and handling storage. There are two types of contamination. First, external and internal contamination. First, external contamination. It occurs microorganisms are present in eggshell surface. Their sources fecal matter from laying hens, soil, dust, feathers and litters, contaminated by nesting materials, improper handling during collection and transportation. Common microorganisms caused by Salmonella species, E. coli, Staph aureus. And effects are microorganisms on shell surface may penetrate through shell pores, increase the risk of foodborne illness. Next, internal contamination. Internal contaminations occurs when microorganisms present inside the egg, like albumin or yolk. These are modes of entry. Penetrate through shell pores, especially in cracked or thin shell. Infection of hens reproductive. Poor washing practices using contaminated water. Common pathogens are Salmonella enteritis, Proteus species, and Pseudomonas species. Effects are rapid spoilage of egg contents, development of odors or discoloration, health hazard to contamination. Factors influencing egg contamination: cracked or damaged cell, high temperature, and humidity. Next, spoilage. Spoilage of eggs are mostly due to their high moisture content and rich nutrient substance. It refers undesirable changes in physical, chemical, and sensory qualities of egg and unfit for human consumption. Egg spoilage is mainly caused by microorganisms entering through the spores, crackings, or during improper handling. There are seven egg spoilage. First, green rot, black rot, red rot, mold spoilage, sour egg, ropey egg, and putrid egg. First, green rot. Green rot is commonly caused by Pseudomonas species. Develop a greenish discoloration due to pigment production by bacteria. Fruity or musty odor observed. The spoilage occurs condition of high moisture and poor. Black rot. Black rot is caused by Proteus species. It turns black or brown color. The strong foul odor is produced due to protein decomposition. It indicates the microbial activity and occurs during prolonged storage at warm temperature. Next, red rot. It caused by Serratia muscens. It exhibits red or pink coloration due to production of red pigment by the bacteria. It emits unpleasant odor in egg, unsafe for consumption. Warm and humid storage favors this spoilage. Mold spoilage. It caused by molds such as Penicillium, Cladosporium, and mucus species. It appears as green, black, or white patches on the eggshell. It may penetrate into the egg contents. It produces a musty odor and results in off flavors. High humidity and poor ventilation promote the mold growth. Next, sour egg. It's caused by fermentative bacteria that produce organic acids. It emits a sour or acidic smell and albumin thin or watery. Spoilage occurs due to improper refrigeration and extended storage. Next, ropey egg. It caused by alkaligens and Enterobacter species of microorganisms, and the egg white becomes thick, viscous, and rope-like due to the slime formation. It indicates the microbial spoilage and enzyme degradation of egg proteins. Next, putrid egg. It occurs due to the proteolytic bacteria that break down the egg proteins and produce gases such as hydrogen sulfide. It gives strong rotten eggshell. Its contents become watery and discolored. These types of spoilage commonly associated with severely spoilaged eggs. Preservation. In this preservation, use of irradiation. Some pathogens can be inactivated by ionizing radiation. There are two types of preservation. First, small scale, next, large scale. First, small scale, dry packing. The eggs are kept in earthen pot with clear dry packing material and pot is buried in wet sand. Immersion in liquid. It is an old method. It prevents of evaporation of moisture from rain. Depending on the liquid, it may inhibit bacterial decomposition by chemical action or physical mix. Next, lime water treatment. Lime water is prepared by mixing about 0.5 kg of quick lime. Quick lime means calcium oxide. It about 1 liter of boiling water. The mixture is left to settle overnight and the clear supernatant liquid is poured into an outer jar. Sodium chloride is added because the increase of specific gravity of water and minimize the chance of breakage of eggs. In this solution, 2.5 liters of cold water is added and filtered through muslin cloth. Keep the eggs to preserved in a glass jar or earthen pot. Pour the lime solution over the eggs till the eggs are completely immersed. The eggs kept into the solution for 24 hours to maximize the beneficial effect. After 24 hours, they are taken out, dried and arranged in filler flats. Eggs can be kept for 2 to 3 months in good edible condition or ambient condition. The only advantage is taste of lime can be detected in the eggs. In next, large scale cold storage. Eggs are selected by general appearance or canning. Chilling preserves most egg shell quality. Temperature between 0 to 1 degree Celsius. Humidity are common 80 to 85 percentage. As circulation in storage home, after some 11 months, a flavor may develop with prevented by periodical sterilization with disinfectant gas such as ozone. Next, thermostabilization. This method is good for fertile eggs since it kills the embryos. It also known as defertilization method. It essentially consists of immersing eggs in hot water at 130 degrees Celsius Fahrenheit for 30 minutes. It tends to coagulate the albumin and the egg is cooled under sterile water. Treated eggs remains edible for 3 to 4 weeks even during the summer months. Though this method has many advantages such as stabilization of albumin, sterilization of eggshell, losses the property of forming to remarkable extents. Embryonic development in fertile eggs is completely arrested. Next, dried and frozen eggs. In this method, the preserving contains edible eggs. Egg products are commercially utilized or prepared by freezing or drying egg method. Albumin flakes, egg and white powder of yolk will be produced by drying process. Frozen egg or frozen egg white produced by freezing method. For egg white powder production, the best in preparation is spray drying, and the for albumin flakes, pan or cabinet drying method is mostly adopted. Thank you.